The genotype is the genetic makeup of a cell, an organism, or an individual, usually with reference to a specific characteristic under consideration. For instance, the human CFTR gene, which encodes a protein that transports chloride ions across cell membranes, can be dominant as the normal version of the gene, or recessive as a mutated version of the gene. Individuals receiving two recessive alleles will be diagnosed with cystic fibrosis. It is generally accepted that inherited genotype, transmitted epigenetic factors, and non-hereditary environmental variation contribute to the phenotype of an individual. Non-hereditary DNA mutations are not classically understood as representing the individual's genotype. Hence, scientists and physicians sometimes talk, for example, about the type of a particular cancer, that is the genotype of the disease as distinct from the diseased. Genotypic and genomic sequence. The genotype of an organism is the inherited instructions it carries within its genetic code. Not all organisms with the same genotype look or act the same way because appearance and behavior are modified by environmental and developmental conditions. Likewise, not all organisms that look alike necessarily have the same genotype. One's genotype differs subtly from one's genomic sequence. A sequence is an absolute measure of base composition of an individual, or a representative of a species or group. A genotype typically implies a measurement of how an individual differs or is specialized within a group of individuals or a species. So typically, one refers to an individual's genotype with regard to a particular gene of interest and, in polyploid individuals, it refers to what combination of alleles the individual carries. The genetic constitution of an organism is referred to as its genotype, such as the letters BB. Genotype and Mendelian inheritance. The distinction between genotype and phenotype is commonly experienced when studying family patterns for certain hereditary diseases or conditions, for example, hemophilia. Due to the diploidy of humans, there are two alleles for any given gene. These alleles can be the same or different, depending on the individual. With a dominant allele, the offspring is guaranteed to inherit the trait in question irrespective of the second allele. In the case of an albino with a recessive allele, the phenotype depends upon the other allele. An affected person mating with a heterozygous individual, there is a 50-50 chance the offspring will be albino's phenotype. If a heterozygote mates with another heterozygote, there is 75% chance passing the gene on and only a 25% chance that the gene will be displayed. A homozygous dominant individual has a normal phenotype and no risk of abnormal offspring. A homozygous recessive individual has an abnormal phenotype and is guaranteed to pass the abnormal gene on to offspring. In the case of hemophilia, it is sex-linked, thus only carried on the X chromosome. Only females can be a carrier in which the abnormality is not displayed. This woman has a normal phenotype, but runs a 50-50 chance, with an unaffected partner, of passing her abnormal gene on to her offspring. If she mated with a man with hemophilia, there would be a 75% chance of passing on the gene. Genotype and mathematics, main articles, genetic programming and evolutionary algorithm. Inspired by the biological concept and usefulness of genotypes, computer science employs simulated phenotypes in genetic programming and evolutionary algorithms. Such techniques can help evolve mathematical in the solution of certain types of otherwise difficult problems. Determining genotype. Genotyping is the process of elucidating the genotype of an individual with a biological assay. Also known as a genotypic assay, techniques include PCR, DNA fragment analysis, allele-specific oligonucleotide probes, DNA sequencing, and nucleic acid hybridization to DNA microarrays or beads. Several common genotyping techniques include restriction fragment length polymorphism, terminal restriction fragment length polymorphism, amplified fragment length polymorphism, and multiplex ligation-dependent probe amplification. DNA fragment analysis can also be used to determine such disease-causing genetics aberrations as microsatellite instability, trisomy or aneuploidy, and loss of heterozygosity. MSI and LOH in particular have been associated with cancer cell genotypes for colon, breast and cervical cancer. The most common chromosomal aneuploidy is a trisomy of chromosome 21 which manifests itself as Down syndrome. Current technological limitations typically allow only a fraction of an individual unregistered trademark's genotype to be determined efficiently. See also, endophenotype, phenotype, potentiality and actuality, sequence, references. External links, genetic nomenclature.